Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Hello viewers, here we are. Welcome back to the next sessions as regards to what we are having here in our home sessions for online learning and broadcast. So today we are looking at, again, Unit 3, as you can check, the chromosomes. And the subject is biology, and we are looking at what is to be covered in Senior 5. But remember, even you those in Senior 6, you take advantage of that, and that's why we are looking at it as a review for you who have already covered this in the previous terms. 2, the lesson we are looking at today is going to cover the RNA. Remember, we talked about the DNA the previous session, if you can remember well. 2, we should also be in position to cover under that section how it is going to differ from the DNA as we look at these structures. Remember, what we talked about last time as these acids that are found in the nucleus of the cell, and we also said that our focus was about eukaryotic cells, and so when we talk of the DNA, we talk of the RNA, we're essentially looking at eukaryotic cells. The objective of the lesson, 1, is that we should be in position to describe. Remember, these command words keep coming up again. Describing, outlining, state, explain. So our guidance is going to be on the description, telling how the structure is designed. Remember, in the description, we look at the composition, we look at the arrangement, and then we also look at the overall structure when all these components are put together. And then we should also be in position to list other forms of RNA. What are we talking about here? When we talk of other forms, in other books, they look at this as types, depending on the writer, kinds, or the different forms of RNA. The last part is we are going to make a comparison, relate. You have the left and the right, so you look at those two and see where components look the same. So the word comparing, therefore, may be taken for similarity. Are we having structures in the DNA we looked at the previous lessons and the RNA we are going to look at today that they share, so that we look at it as common characteristic in all of them? But also make a contrast. This simply means a difference. Are they differing? And if they do, how? If they are all nucleic acids within the nucleus of the cell, how different are they? So that's our coverage today. So here we go. I want you to take some bit of time and internalize this so that you know that by the time our lesson is over, we are able to capture these. So we start. The ribonucleic acid, which is abbreviated as RNA, as you can see, has just a few of short descriptions we would look at to be able to give what we are expected to capture at the end of the lesson. 1, that it is a single. So it is 1 nucleotide, which takes us back to what we were looking at in the very first lesson when we talk of nucleotides and what a nucleotide actually looks like. So where we come here and find out a component and structure it out, as you can check here. If you get close enough, you should be able to see the label as 1 nucleotide. So it means this component, as you can see the way it makes a demarcation of one of the structures along here, in which we have a P, we also have a C, we also have an S, where S stands for sugar, P stands for a phosphate, and then we would have this as our organic base. Maybe in this case would be cytosine, as you can check one here. So remember, we said that the DNA is composed of nucleotides as well, and so is the RNA. But where is the difference coming from? Check this out very carefully. So we'd have this as our DNA. So I'm putting it here for us to be able to remember what we looked at in the previous lesson, because at the end of the day, we need to be able to make a comparison. Remember, that's very important. So you have your DNA here, and now we are looking at the RNA. One, single nucleotide chain. So it's one word, single nucleotide chain. And they're saying it's helical. The structure is in a spiral form, and so is the helix. So we come here to these structures, and then look. If this is our DNA, look very carefully. And because we say that the DNA is double-stranded, so two strands, while here they are saying it's one chain, a single nucleotide chain. So here we come. If this is our DNA, and then we zoom in, we get close enough, so that we have this, which is detailed here. Check very carefully. Double-strand DNA, stretched out, zoomed in to check the components. Then we have two strands, and here we go. These are the bonds we have been talking about. So you have the sugar, a phosphate, a sugar, a phosphate. So they go alternating, like we said in the previous description, alternating units of a sugar and a phosphate. Alternating units of a phosphate and a sugar, a phosphate and a sugar. And then in between are the other sections of the ladder we talked about. The pair of bases, in which we said we have the large ones and the small ones. Single ring, double ring. So by comparison, therefore, this is expected to be bigger than this. Two rings, one ring. So small base, large base. Large base, small base. Now when it comes to the end, the RNA, here we go. Check it out. Two chains coiled around each other to form a double strand. A single chain. The bases are paired, and here they are not. I hope you're looking at it very carefully. We check again. Two chains DNA, single chain RNA. Second point, they're saying that it has a pentose sugar, a ribose. Interesting point we need to look at now, a ribose. But in the DNA, we say deoxyribose. Now the question comes in, where does the difference arise? Which takes us to this. So let's get close in. Let's get close here and check. If we say a pentose sugar, yes, it's a 5-carbon sugar. So we count 1, 2, 3, 4, and 5. So those are 5 carbon atoms on the sugar. 5 carbon atoms, pentose, 5 carbon sugar. When it comes to this same sugar here, we need to check very carefully. So I want us to zoom in very well and check, one, the names given. A ribose sugar, and so here it is, under the section of RNA, deoxy, ribose. They are all 5 carbon. but the difference in the words is here, while here there isn't. So we trace it within the atoms in arrangement. So let's get far deep inside and be able to read out. So you should be with me here very well and check. OH, OH. So on carbon number 1, then carbon number 2, and carbon number 3. So at C3 and C2, under it, you have this. This is our ribose. Deoxyribose. We have H, no O, and OH. So it means, therefore, that the difference lies in how many hydrogen atoms we do have, or how many oxygen atoms we possess. On the third carbon atom, on a ribose sugar in RNA, you have OH. On the third carbon atom, on the deoxyribose sugar, we have OH. But on the second, so which is this, we have O. Here we have only H. So it means, therefore, that one structure is less in terms of oxygen atoms by 1. 1, 2, 1. So we shall say deoxy, while here we say a ribose. So it has one more oxygen atom along this position. That's where the difference comes from. Third component of the RNA, that it has a single nucleotide with a base either A, or it can have a C. So we go back to the words in which we said this was adenine. And then you have this as cytosine, like we did before. Then here you have guanine. Now, something is happening here. We are missing the usual letters represent these bases we've been used to in DNA. Here, T, adenine, cytosine, and guanine in the DNA. When it comes to the RNA that is stretched out of here, which we now have brought down here, so this is my zooming in. I'm expanding this to here. That each of these letters are being represented here. You will check. We have a U, you have an A, you have G, you have C. The same applies here. A, U, C, G, A, U. No matter the arrangement. If they tell you to draw a structure, you don't need to remember exact arrangement of these letters, as long as, rule 1, that you have uracil instead of thymine. So it means that a U goes for a C. That's the replacement we are talking about. It therefore means that, like we said in DNA, 4 types of nucleotides, here also in RNA we shall have 4 types of nucleotides. But the difference shall lie in the substitution of letter T in DNA for U in RNA. That's why we've written them here and say this. It means it can be this on a nucleotide, this on another nucleotide, this on another nucleotide, or this one. So we have 4 types. Which takes us to this detail. Don't mind about the arrangement of these atoms in this detail here. You can either have it this way or a simple representation as we do have. So that we have this arrangement. As you can check, remember, these bases differ. in size. So here we go, a large one. This is a large one. Then you have a small one, and this is a small one. Remember that this substituted a T in the DNA. All right, so here we go. While we already have our structure of RNA in which the base is uracil instead of thymine, very important point, a sharp difference you need to always remember. 2, that the sugars differ. So the major differences between this DNA and RNA will lie under 1. Which base do you have? Is it a T or a U? And secondly, what about the sugar? So we have the bases here, thymine and uracil. Again, I will still emphasize at your level, you do not need to understand the molecular arrangement here for these atoms to have that. As long as you know that larger bases, double ring organic nitrogen bases, small bases, just a single ring, as we have been showing you here in the DNA. So this is your RNA with these features. Here you have it, a sugar alternating with a phosphate. A sugar alternates with a phosphate. Sugar, phosphate, phosphate, sugar. Then this is our section where the bases are. Now a single component here is what we shall have as our single nucleotide. I hope you're seeing it very well now. But we're saying it's a chain. So it's a repetition of many of these. That the first will differ from the second because this has C and this is G. The second differs from the third because C is first, G is the second, third is for A, and then the fourth nucleotide, again, 3 components, a phosphate, a sugar, a ribose sugar, and an organic base. Now, look at this again. This is small, and this is small. So we go back to the other words, the purines as the large ones, the pyrimidines as the small ones. So here we go. Now, this is essentially what the structure of the RNA is. A polynucleotide composed of small units called nucleotides, in which you have an organic base as U instead of T. Second, we can sum up here as under that objective that it is single strand, as you can check. It is a single strand. It is a single strand. And contains the sugar as a ribose, a 5-carbon sugar with 2 oxygen atoms above that of deoxyribose by 1. So, third carbon OH, fine. Second carbon atom, you have OH, here you have only H. So that's the difference. Then we do not just have, if this is just a typical structure, if we have the RNA, but does it have forms? Remember, it's DNA, but when it comes to RNA, then there are variations. We have varieties, different types, in which we get the names of these forms depending, one, on the structure, the composition, and what we are going to do. Remember, change in structure in most cases, you do have a new function coming up. So if you look at this very well, that messenger RNA, there's one thing I want you to pay much attention to, that you have the abbreviation is written in a way that the word messenger is represented by a small m, transfer represented by small t, and ribosomal represented by small r. So these are the abbreviations, mRNA, tRNA, then ribosomal RNA as rRNA. As we would look at this in terms of structure and then design and function, we can also attach the names to what they do. So if we look one, messenger, so it carries the message. Transfer, there must be some bit of transport. And ribosomal, this, if you remember your cell structures, these are cell components. So the RNA found in the ribosome is actually called rRNA. tRNA will transfer. We shall look at these in the next lessons that follow during protein synthesis and DNA replication. Then you now have these variations having mRNA, tRNA, and rRNA brought about by changes in shape. 2, the length, how long it will look, depending on how much message it will carry, which will also connect us to what they perform. Like I just said, transport, proteins, as we shall see, amino acids, carry a message from the nucleus to where it's needed for processing, and ribosomal RNA as in terms of where it is located and what it's going to perform. That's well ahead in the next lessons that come afterwards. So, looking at what we have just seen, you should be in position to make a contrast. We looked at DNA, and we have an overview here, which we looked at last time. We've also looked at the RNA and its descriptions, as well as structured here as some bit of where the differences come. Is it the base? Could it be the sugar? Think about that. So if I'm to make a contrast, what exactly do I have to write? Simple. The differences. So we shall look at the differences based on composition. So if I say I'm going to consider the base, says, and 2, I look at the sugar present, like we just talked about, and 3, I also look at the size. Can I maybe say mass? For those who do take chemistry as well, when we talk of molar mass, what are we looking at? Which is very simple. Let's look at this. If this is a double strand, and this is a single strand, it's simple math and comparison. If I made this a single strand, then it means that one component would be half the amount. So if I'm talking about mass in terms of, I'm going to look at which one is half the mass of the other. Can I think about that? If it is the sugar, am I looking at a ribose against a deoxyribose? You need to think about that as well. If they are the bases, am I looking at a T replacing a U? When we talk of the comparisons, we are simply looking at how do they look the same. Are they all composed of polynucleotides? So you need to think about that. 2, do they all have an A? Do they have a C? Do they have a G as bases? Are they all genetic material? So when it comes to the activity we are having here, they just want us to list the components of the RNA nucleotides. So we go back here and pick out a nucleotide that is for the RNA, based, remember, on the description of a nucleotide chain in the RNA. So where we now look at, are we having a sugar? But if it is the sugar, is it a ribose? Then we have to indicate it, because we need to make a sharp difference between the RNA and DNA. So we make no mistake here. 2, when we talk of organic base, what do we consider? 3, the phosphate group, as we've just seen in the structures. Now they are asking us, if we have listed these, how will they differ from the ones that are meant for the DNA? Is the question here as an activity taking us back to what we've just seen? Can we use this as a basis to be able to answer the question? So when they say how, we need to show. So we give the components and how they bring out the difference. If a DNA nucleotide is composed of a T, at least one of them, what shall be for the RNA? Shall we have a U? When it comes to the sugars, are we in the RNA looking at it in terms of a ribose? That's what they want us to look at. So here we have the third question. I hope this is very clear. And saying, how are the nucleotide components held together? So you have these, and we need to assemble them. The stake is 3 marks. So it means, therefore, you need to think, if this was taking 3 and I'm listing, this one is taking 2 marks, how much? Why is it that we are being asked to give extra? The stake of the question will determine how much you write. So if we look at the combination and joining, then we would have this. I want you to look at this very carefully. Sorry, I did poorly. And then you have another component. So if I extract this, I would expect to pull out water, and the same applies to this. I would pull water out. Now, if all this is got out, then we have what we refer to as condensation reaction. So maybe one may think, could the question they are asking us, requiring us to show how water is removed, and 2, by what process, and finally, by what design, so that at the end of the day, we actually have something like this. So we look at this taking us here. Sorry, let me use another marker. And this taking us here. So we have this as a phosphorester bond. And then having this as 1 plus 2 forming our nucleoside. So we get a nucleoside and we add a phosphate group, and then we form our single nucleotide. So with that, we come to the end of our session, and I hope we have been able to capture all these, describing the structure we have done here, and comparing, listing the forms of RNA, and 3, comparing and making contrast between the 2, and in position also to have a recap using the activity as we have seen. Thank you so much. If you have some time to capture some of these images, the time is yours now. Thank you.